Hi, everybody. My name is Lucine Devejian. I'm a member of Dr. Mandal's lab in the Department of Chemistry and Biochemistry here at UTA. I'm an undergraduate senior, and my project is about the effects of resveratrol on neuroinflammation. Neuroinflammation is one of the main causes of neurodegenerative diseases such as Alzheimer's, which currently affects over 6 million people but has no cure. Resveratrol is a naturally occurring polyphenolic compound, which is well known for its antioxidant, anti-cancer, and anti-inflammatory properties. And the goal of this study is to test resveratrol as an effective inhibitor of inflammation in mouse microglial cells. I used the BV2 cell line. So what we did to perform the study is we took the BV2 mouse microglial cells, treated with resveratrol, followed by lipopolysaccharide, or LPS, in order to stimulate inflammation in the cells. And then we collected the supernatant and the cell lysate. And from the cell lysate, we isolated the RNA, synthesized cDNA, and ran PCR as well as real-time quantitative PCR. And then the supernatant, we removed any cell debris and performed the supernatant-based analysis. The first thing we performed was a regular PCR, and we measured cytokines such as IL-1 beta, which are known to be upregulated upon inflammation. And as you can see, IL-1 beta was significantly decreased with the addition of resveratrol at 10 micromolar compared to LPS alone, indicating that resveratrol effectively reduced the neuroinflammation. We turned to real-time qPCR, where we tested cytokines, again, such as IL-1 beta and also IL-6, and we determined that compared to LPS alone, resveratrol was able to significantly reduce the levels of these cytokines in the cells, again, indicating that inflammation was reduced. Again, also with the supernatant, we measured nitric oxide levels, which is a biomarker of neuroinflammation because macrophages release nitric oxide in order to fight infection and bring down the inflammation. And we were able to see that resveratrol successfully decreased the levels of nitric oxide in the cells, which further supports the idea that resveratrol is bringing down inflammation. In the future, we plan to extend these studies to protein and measuring how resveratrol is able to reduce the levels of various biomarkers in the NF-kappa B inflammation signaling pathway to see how resveratrol is working on the signaling level. We also want to test long non-coding RNAs that are known to be upregulated during neuroinflammation, which are also important biomarkers of inflammation and neuroinflammation. And finally, we also want to extend these studies from mouse cells to human cells using human microglia, otherwise known as HMC3 cell line, and to use A beta to induce neuroinflammation instead of lipopolysaccharide, because A beta plaque formation is one of the causes of Alzheimer's disease, whereas lipopolysaccharide is a more general induction of inflammation. We believe that the future findings could determine whether or not resveratrol is a potential therapy against neuroinflammation and neuroinflammation caused diseases. Thank you for listening.